Hi, Mr. Khademi. How are you?
I'm
Uh,
good. How are you doing?
Good, good. Thank you. Um, so we are here for an interview to ask you some questions regarding research and your field of expertise. So we'll get started right away. So what do you think, what does like a technical day or week look like for you as a researcher?
So, I think the beauty of doing a PhD is that there isn't necessarily like a consistent schedule that you're really following, but I think the way that, it's generally set up is so let's say I'll go into lab in the morning and you just start with checking up on all of your experiments or reagents that you've consistently used. So, for example, I have cells in culture that I have to look at, make sure they're alive, make sure that they're growing. Um, and I also work with mice. So, I'll go check up on some mice. For example, there's some mice that we inject with cancer cells. So, we go check up on them, make sure they're not dead yet, check, the size of the tumors. And once I get a sense of like, okay, this is how everything's looking, then I'll essentially have like a planned list of things that I need to do that day. So basically, I'll be a mixture of like both at my bench doing experiments with my hands. So that's going to be wet lab work. And then if I'm waiting on stuff or if I don't have any wet lab research to do, then I'll just be at my computer, either, you'd either be reading papers or you'll be analyzing your data. But I think what encapsulates what a research program looks like more consistently is that you're basically you basically have like a set number of things that you want to do on like a regular basis. So let's say I start off January saying, okay, so this is my goal for this month. I want to answer these questions about, a sub part of my thesis, and this is what I have to do. And so you can generally plan out, but the way it works in research is that there's no real timeline that you can follow because you can't predict what's going to happen. So that's why it's very flexible and you have to be very much on your toes. But that is what I
Yes.
would say.
So I think like a misconception is that I think like a lot of people think PhD is taking like a maybe courses that you have to do, or you're still doing what you do in bachelor's where like as an undergrad you're taking exams and whatever. But from what I'm hearing is that it's probably more flexible than that, and you're like doing stuff that what you want to do more than like what you have to do, you know.
Yeah, yeah. I mean, I guess like there's definitely still stuff that you have to do, but you are working on stuff that you want to do. We still take courses though. Every I guess PhD program, depending on the school, has its own structures. For example, for us, we had to take all our courses in our first year. And so we just get that over with. So it's like an extra semester of undergrad. And then we basically just do research. Whereas I have friends in other research programs who are taking like one or two courses a year. So they're still actively taking courses. But it is very flexible in terms of like you decide your schedule as long as you're getting things done.
Oh, okay, I see. And so the second question that the students had is, what skills do you use most often in your work?
What skills do I use most often? I think there's both mental as well as like actual technical skills that you need to have. I think when it comes to mental, one of the most important things is just a sense of resilience, is just you have to constantly face failure. And so being able to internalize that failure and essentially see it as not a reflection of you, but as a reflection of, you had a really good question that you asked about, biology. And then when you do the experiment to answer that question and it doesn't come out the way you want it, it's not a reflection of you, but rather just a reflection of biology. So being able to compartmentalize what failure means is really important. It's something that even I'm not good at 5 years into my PhD, right? Like, it's very difficult coming from, undergrad where you study a textbook. So the answers are there. You just have to answer questions on an exam, right? They're tangible, actual outputs. But in research, everything is uncertain. So being able to compartmentalize. I think critical question and analytical skills are very important because it's no longer like you're just doing things. You have to do things knowing why you're doing them, because you're constantly going to be asked questions, you're going to be giving talks, um, and so having a critical understanding of what you're doing, as well as being able to explain your research, is very important. And so, um, yeah, just knowing what you're doing at a critical level as opposed to just like surface level, oh, I'm doing this experiment, but I don't know why. Um, and then I guess other skills, I think very much adaptability, because you're constantly going to be learning new things. You're in a field where you're not going to be repeating the same thing over and over and over again. So you'll do an experiment and then something interesting will come out of it. For example, if you're studying, let's say, cancer, right? You're studying leukemia, and then all of a sudden you get something that's related to immunology. If you know nothing about immunology, you'll have to learn it, and you'll have to learn the techniques associated if you want to follow that. So you have to be very adaptable in your thinking. And I think that also includes being adaptable in terms of like shifting your research project, because one misconception is that, you start off your PhD with a question and that's the question you're going to write your thesis on. That's very much not the case. You typically start with something and you end with something else, because research programs evolve. And so being able to adapt and not get very attached to a hypothesis or like your project is very important, because what's likely is that nothing interesting will come out of those initial questions. And so you have to be willing to transition to another project. And then I think just technically you have to be comfortable with, working with the reagents that you have. For example, some people don't like working with mice. They don't like the smell. They don't like how it feels. So you'll just have to essentially get used to certain technical skills. But I think mentally there's a lot more challenges that are employed and people should be aware of before they start.
Okay, I see. So like it's more of like being able to adapt as well, because I think a lot of the misconceptions, also the fact that you start off with one question and then you carry that through all of your PhD, and
Yeah.
it doesn't work like that, right?
Some people do get lucky, and I think like that's like the best case scenario, is that like the first question you ask is essentially what you end up with. And those are the people that end up like, not necessarily finishing faster, but they go through less of a headache of trying to find something. But yeah, like you said, the most common case is you start with something, you end with something else.
Yes. So the third question that we had is, what educational background is required to work in your field? And
Like post PhD or getting into a PhD?
well, for both.
So to get into a PhD, I think the most important thing is that your background, so whatever like major specialist you studied, is actually relevant to the field that you're applying to. For example, like my advisor is on a admissions committee, and they were talking about how they were going through a bunch of applications for this year's like cycle, and there were people that had like background in like dentistry, or like they went to veterinary school, and it's like those are the people that just get eliminated immediately because they absolutely have no relevance. So their experience has nothing to do with it. But for example, if you studied something like chemistry, or if you studied something like engineering, these are closer to what, for example, a cancer biologist would work in. And so essentially anything in STEM would make you applicable to study for it. And I think that's another good thing about a PhD, is that you can have a diverse background, or a very different background, but somewhat still related. But for example, I studied genetics in undergrad, and so that was very much relevant. So that's why like the link is there. But I think when it comes to after, I think this is another misconception where, some people think that you can only really go into like academia. You continue as like a postdoc to then become a professor. But there's like a plethora of different career paths that you can take. And essentially you have to cater your experience. So that becomes a totally different question of like what experiences you need as opposed to your background. But yeah, I think that would answer it.
Yeah. Okay. I see. And for the fourth question is, what should students start doing early if they want to pursue research in the future?
So assuming that this is like a high school student, an undergrad student,
Yes, like
what
more
what
of like
stage?
a high school student, yeah. Like for students who right now in like grade 9 to 12, like what should they be doing? Like,
Yeah. I think definitely focusing on your high school studies for sure. I think it's very rare for a high school student to get engaged with research programs. I know that there are some schools that offer, for example, like research programs for high school students. The institution that I went to, or the high school that I went to, didn't. But I think the best thing that you can do early on is gain a sense of understanding of what research really means. So essentially understanding, you know, like you said, what a typical day looks like, what a research program actually looks like. Because for example, when we read a textbook and, it makes a claim that this protein does this thing. So this protein is a tumor suppressor. That statement does not capture like the years or decades of research that like is actually behind making that claim. And so understanding like what the process of starting with a question and then making that claim is very important, right? So essentially I would say very early on understanding, what research is. Because everyone knows what, for example, it's more common for people to know like, you know, how to become a doctor. You do your undergrad, you take the MCAT, you go to med school, and then a few more steps and you're a doctor. But no one really tells you, what the path to a research program is like and to a PhD is like. So I would definitely say, read into the basics of, what a PhD program even is about, what research entails. And then I think once you're an undergrad, I think the most important thing is like actually immersing yourself in research, right? You become an undergrad researcher, you get familiar with failure, but also just like having your own research question that you're helping to answer, and then just doing experiments to fail because no one really prepares you for how much you're going to fail in grad school.
yeah. Okay, that's very true. And for the fourth question, we said, what should students start doing? Oh, yeah, sorry. Yeah. What experiences mattered the most when you're getting started with research? Like, what did you think?
So, what experience that I had.
Like early on, like what did you think like helped you when you got started? Like, for example, before you did your PhD, what experiences led up to that moment where like it helped you? Like for example, if you were an undergrad and like what did you do in undergrad that helped you later on when you pursued your PhD?
I think it's one of the skills that, I had in undergrad and then I continued on in my PhD with that was very helpful is trying to fill in the knowledge gaps. So for example, if like on my lecture notes it said like, again, let's say like it makes a claim that like this protein or this gene is a tumor suppressor. And then it'll list the ways that it functions as a tumor suppressor. If something in that explanation doesn't make sense, it's trying to really dig into all the little details that add up. So it's just like not letting any confusion slip through the cracks. Because once you get into a PhD, you get into this like really critical level of thinking where even the smallest thing can essentially negate your hypothesis or your project. And so it's really critical to understand what these minor points are and essentially understanding everything as a sum of its part. So essentially, if something isn't sticking, constantly iteratively ask questions until it sticks. And I think that's one of the most important skills that a PhD student can have is understanding how to be critical about, you know, um, about anything really, because you're going to be reading like so many research papers and so many research articles, and you need to be able to know what questions to ask. If they're making a claim, you can't just trust the claim, right? Because like, let's say you take that claim and then you want to ask another question about it. You need to make sure that that initial claim is like very strong in order for you to follow up experiments, or else you're going to be wasting months or even years of your time. So definitely that level of critical thinking. And again, I can't emphasize this enough, but being comfortable or more comfortable with failure and knowing how to reframe what failure means, right?
Yeah.
Failure in, high school or in undergrad typically means that like, oh, you didn't do as well on your test as you wanted to, or um, it's all grade centric. But a failure in grad school becomes like you're doing an experiment like 10, 20, 30 times, and each time like something goes wrong, and you need to be okay with that process and understanding that it's a requirement, right? It's not as big of a barrier as getting a bad grade.
Okay. So like that could be like a really good lesson for like people who are even in high school where they're worrying or stressing about the grades and like being more comfortable with failing. Not in terms of like you should do bad in school or anything, but like being more comfortable learning and also making mistakes can help you later on in life when you
Yeah. And I think
research.
the most similar experience that anyone can go through is obviously like you said, like do well, because like you need to do well in order to advance through like, undergrad and then through grad school and med school, whatever post undergrad you want to go into. But I think like engaging with, for example, a new hobby, right? Something that is low stakes, but there's enough of a skill curve that you're gonna fail throughout this process. And I guess the best way to summarize it is just like, don't expect to be an expert from the start. And so any experience that allows you to understand that will help you. And so, for example, if I wanted to start to learn crocheting, which I don't crochet at all, I would start and then I'll see how uncomfortable it gets when I'm learning something new and I'm not very good at it. But, through iterations, through different, I guess like by just sitting with the hobby, you eventually get better at it, but you're not good at it to begin with.
Okay, I see. And for the next question is, what happens when funding runs out in like a research firm?
This is really institution dependent, and which is why it's important to know, before you apply. It's like, what are the fail safes of these different institutions? For example, in some cases, the lab completely shuts down without any sort of help from the school. And so the graduate students either have to relocate if they're able to another lab at the institution, or they just masters out of the program if there's no other choices. Because typically transitioning from one PhD program from one school to another schools is more difficult. There's more hurdles to get through. But at certain institutions, the school actually has this amount of money or finance stowed away that, let's say, if a lab doesn't get any funding, the school is going to allocate some of that money to the lab as essentially like a buffer for that lab to get more funding. I think this is something that's become particularly relevant now, at least in the US, where there's been budget cuts to the research programs. And so a lot of labs have been very careful to spend less money. And some labs have had to shut down. They don't take graduate students anymore as well. So yeah, it's very much institution dependent. And it's something definitely to look out for when you're joining a lab is, how much funding does your advisor actually get? Do they have a good track record of making, successful grants to bring in money?
Okay, I see. So that is something that you have to actually research before you go
Yeah.
into it. Yeah. All right. And the next question you had is that what are the key differences between academic research and industry research?
So one of the key differences is the amount of flexibility that you have in academic research, because you're generally involved in, one big question, right? So for example, our lab studies gene regulation, right? And cancer. We can really study anything that falls under that umbrella. But when it comes to industry research, you're very much, let's say, committed to a particular thing. Let's say I'm working for a company. I don't have the leisure of studying what I want. I have to study what they want. And typically what companies want is to make profit. And so that includes drug discovery or drug validation or creating any sort of device that has some sort of actionable therapeutic use. And so you're much more restricted. But you're a bit safer in that you don't have to worry about getting grants, right? Because the company is just going to make, just like any company, right? It's going to work for profit, and that's how it's going to pay its employees. So it's a bit safer on the finance aspect. So yeah, one of the biggest differences is, I would say, is the flexibility and the questions and the work that you're able to work on. But yeah, that's
That's
the
it.
biggest
Okay.
difference. Yeah.
Yeah, so it's more about like the flexibility and also the amount of money you can get through the industry as well, right? Because they have more finance in there. So,
Right.
and the next question that we have is, what mistakes do you think young researchers often make when they start?
Can you repeat that?
I say, what mistakes do you think that young researchers make when they start?
Ooh, I think this is something that I don't know how common it is, but I've seen it in some cases. But I think when it comes to, for example, undergrad researchers, or less so like PhD students in their like first year, when you first start a project, a lot of undergrad researchers just want to do the experiments that they're told without really thinking about why they're doing those experiments. And I think one of the biggest mistakes you can make, therefore, is not really engaging with the why of what you're doing as opposed to just doing the what. So understanding like the technical aspect of the experiment that you're doing. For example, if I'm doing a Western blot, I have to understand what a Western blot entails. What am I looking for? What are what are the specifics of this technique? And in this way, you're not only, training yourself to be a critical thinker, which is not something that's unique to just a PhD, but to any career path you want to go down. Understanding each part of the thing that you're doing will not only increase your expertise, obviously, but it'll enable greater troubleshooting and a greater understanding of the results after. Furthermore, I would say related to that is not understanding the specifics of the project. So again, if I'm doing a Western blot to look at the expression of a particular protein, I need to understand what question I'm even asking, such that once I get the results for this specific experiment, I can then start to theorize about what's going on and what follow-up experiments I need to do, right? And I think the mistake that people make here is that they'll be told to do this experiment. They'll do this experiment, but then you ask them a question about like the results or, follow-up experiments, they won't be able to answer because they just know the technique that they did. They just did it out of, let's say, habit, but they can't explain anything about it. And so I think that would be the biggest mistake, is just not engaging with the project's details enough.
Okay, so it's like you have to know what you're doing essentially, like, any
Exactly.
experiment. Yeah, okay, I see.
So knowing the hypothesis, knowing, the purpose of this experiment, what are we testing for exactly? Because if you don't know what you're testing for, or if you don't even know the question, it's just like, how are you going to analyze the results? You don't want to be just someone who does the experiment. You want someone who can drive experiments forward, drive projects forward.
Yeah, well said. And another thing we're going to ask is there anything you recommend that, students should read, learn, or work at this stage? So like, for example, what separated successful MIT students from average ones in your experience?
So I think first and foremost, something that I said before is reading about what a research program even looks like what even does a PhD entail. And I think there's a myriad of resources of like people explaining, their day to day if they can, like watching videos of PhD students, like vlogs, of them going into their lab, doing experiments, reading any sort of source. I'd say you can try to read research papers, but I think those might be too high level and you might not be able to get as much. But I think if you can read an abstract, so just like that initial summary of the paper, and see like what even is that they're saying, skimming through the paper, seeing like, oh, let's look at all these figures, let's look at all these like different sections. There's a surface level of understanding that comes with that in terms of like what level of detail is required in research, or what's like the stepwise process. Because I think another really cool thing that people don't know about research is when someone presents like a research presentation or like writes a paper, you see this like story of them saying we started from point A, we went to then point B, point C, point D, all the way down to like our final point Z. But what you don't know is that when they were actually doing the experiments, they went from point D to L all the way back to B, and then from B they went to, let's say, like X. Research isn't linear. Research is completely you're going on tangents, and it's something we call revisionist history, where you do like, let's say, a set of 50 experiments, and then you put those in order of what you think makes the most sense. So that's another cool thing about research, is that it's not entirely linear. But yeah, definitely just gaining an understanding of what, a PhD actually is, and then what a research program entails. And then I think another good exercise that people can do is essentially you can just be aware of like I said, when you read a statement, right? Like you're in biology class and it says like the mitochondria is the powerhouse of the cell, right? Thinking about like what needs to go behind that? What does each word in that mean? Like what does the powerhouse mean? Like how do we know it's of the cell and not like of, a whole system? I think just like trying to gauge out like if you had to devise like a set of sub questions to ask about that claim, what would it be? So like, is the mitochondria, like if the statement is like the mitochondria is the powerhouse of the cell, it's just like, well, how do we measure the amount of power exerted by something? How do we even know, this is just like theoretical, but how do we even know that all cells have a mitochondria? So basically just like going through the exercise of asking what we would need to know in order to answer that. Because I think that's one of the greatest things about a PhD is that you are essentially answering questions that no one else knows. And so you're sitting with uncertainty, and you're kind of like bringing clarity into the world. You're bringing new knowledge into the world. Because like every single technology that you see that we use today, even something as simple as like a light bulb, the amount of like research that went behind actually creating like a light bulb is crazy. It started with people asking questions just for the sake of asking a question. And someone was just like, hey, why don't we use this person's research, which when they were doing it had no immediate like translatable output, and try to like make something better off of it. And so that's how research just like gradually builds up. And so definitely being able to ask questions and finding ways to ask questions is very important.
Okay. And like also, what do you think is like one misconception that people have about leukemia research that you have learned is wrong?
So I think nothing that we've already said would be new. So definitely like research is not linear. You're jumping from different points. Research is also not starting with question A and ending with question A always. And research is just not easy because when people make, a certain claim, you read a textbook, you're reading all these sentences, it makes it seem almost arbitrary how we came to that, or even trivial. But I think the amount of work that went into like even making a single claim is enormous. So I think understanding like how much work actually goes into discovering something is a lot.
And like just like specifically about leukemia research, which I think is
About
big.
leukemia
Yeah,
research
about
specifically.
leukemia research. Yeah, yeah.
I don't know of many misconceptions that people make of leukemia research, unless there's any that like you can think of. At least there's none that come to my mind. But I think when it comes to anything, so I think like one thing that I can think of is, well, twofold. One is that when people say we found like the cure to cancer, we found the cure to leukemia, those statements are typically just clickbait statements, because leukemia isn't just one disease. It's an umbrella term for like, you know, so many different subtypes of leukemia. So you have like an acute myeloid leukemia, you have acute lymphoblastic leukemia, you have chronic myelogenous leukemia. This is just to say that like there's different kinds, and so there's not one fix all solution to all of these. So that's definitely one thing. And so when you engage with research, you're typically engaging with like one specific type of leukemia, but sometimes you're able to apply what you learned in, subtype A into subtype B and subtype C, etc.
Yeah.
And then I guess the other misconception that I completely forgot while I was explaining,
Yeah.
if
No,
I remember, I'll mention it.
Yeah, no, it's just like even like what you said is that a lot of people I think have the misconception of like thinking that, for example, you see videos online or you see like websites saying, oh, researchers have found a cure to leukemia or whatever. Like it's not like that. It has to be like specific. So like even like when we're teaching students, or like teachers are also teaching students, they should be like specific as well. Because I think
Exactly.
and even when researching as well, I don't think so we're ever taught that like what we're supposed to look for. So because there's a lot of websites that can tell you a lot of information, but knowing which one is like accurate and also telling you the right thing is important as well. So I think it's a great thing that you said there.
Yeah.
And so, and the last question that we have about about leukemia research here is that what problem in leukemia research do you think is underrated but could be huge in like later years to come, like 10 years, let's just say?
Yeah, I think this is something that I would definitely apply to other cancers as well. But I think one of the most important things that, I get to witness like in the lab, because there's like other labs working on it, is that the amount of technologies that are allowing us to further advance what we call precision medicine, which is basically saying that, let's say we have 10 different patients with a specific kind of leukemia, right? So let's say they have, well, let's just keep it at leukemia for the sake of simplicity. So let's say we have 10 patients with leukemia. The cause of each person's disease is going to be very different, right? So let's say person A has a mutation in gene A. Person B has a mutation in gene B, but they ultimately all come down to having leukemia. So there's many roads to Rome, many roads to leukemia. But because they each have a mutation in a different gene, then the treatments or the therapeutics that we provide to each of these patients is going to be different. Some person is going to respond to a particular drug, but then another patient who also has the same leukemia isn't going to respond, because a lot of the therapeutics that we provide, some are what we just call chemotherapy, which historically it was called that because it wasn't known to have a particular target. But for our sake, let's say that it targets a facet of cellular biology that covers almost every leukemia patient. But there are certain therapeutics that target a specific mutation or a specific protein. And so let's say that patient A, their leukemia is driven by protein A, patient B is driven by protein B. If I give a drug to both of these where I'm targeting protein A, only one of them is going to respond. So not everyone responds the same. So we have a lot of technologies that not only allow us to identify what specific mutations or what specific cancer drivers each of these patients has, but what we also have is, let's say that there are mutations that we've not yet seen in any patient, right? So let's say we have 10 leukemia patients and they all have mutations in like 10 different genes. Let's say that there's an 11th gene that we haven't studied yet. What we can do in the lab is we can actually create those mutations in, let's say, mice or in cells, and then we can model or we can test how that mutation can be treated, right? We know that that mutation will cause leukemia, but are there like drugs that we can use? Are they resistant to certain drugs? such that, let's say patient 11 comes into the clinic, we see that they have mutation 11 or this 11th mutation, this 11th gene, we can now treat them as opposed to being like, we have absolutely no idea what to do. Because one of the biggest issues or one of the biggest concerns in any sort of like therapeutic setting, so when you're treating a cancer patient, is that we're right now just giving any drug that we can in order to cure the cancer. But the issue is that more isn't always better. And in fact, you're probably going to cause more harm in the long term because a lot of these drugs are targeting important processes such as like DNA repair, transcription, translation, things that all your cells need, including the healthy cells. So if we can limit the number of drugs that we give to a patient by only giving them the drugs that they actually need because they have the relevant mutations, that's very powerful. So that would be the first thing. The second is at least our institute has a lot of cutting edge research on immunology. When people typically think about, the immune system, they just think about people that, I get a cold or I got the coronavirus. How is my immune system really helping that? But what's really cool is, how we get vaccines to, prevent the flu, or we can prevent, whatever sort of, like babies just get a bunch of different vaccines for a bunch of different viruses. We can actually create vaccines for cancer, right? And so these could be either vaccines that you give beforehand so they can be preventative, like how we do with the flu every season. There's a new variant of the flu, so we get a new vaccine. Or let's say that a patient has leukemia or has any sort of, lung cancer, breast cancer, whatever, we can give them a curative vaccine. And so there's a lot of research being done in terms of the efficacy and the ways to create the best vaccines to treat these patients. So these are two avenues that I would say are very exciting, and not only leukemia research, but almost any cancer research in general.
Yeah,
And there's more, but yeah.
Mhm. Yeah. And would you say like also in MIT, this is how you guys do it, or is it like other institutions are also doing it the same way or not? Because I think this is special towards, the institution that you're in as well, right?
Yeah.
That is.
So I think I can't speak to other institutions as much, but I think what makes MIT, so our biology department is like broken up into different buildings. And so the building that we're at is known as the Koch Institute for Integrative Cancer Research. And the integrative part actually comes from the fact that we house both labs in pure biology, but also in engineering. So we have bioengineers as well as, or I guess like the bioengineers also includes like a lot of chemistry based research. And then you have, pure cancer biologists such as us. And there's this intersection where, let's say, the bioengineers will create something. So for example, they're going to create special formulas that we can use in vaccines, and then we can test out as the pure cancer biologists. So I think it's that integrative approach that allows the interface of engineering and pure biology to come together to create, strong research programs, but also like very actionable and translatable research.
Yeah. Okay. That's very interesting. I actually didn't know that.